All right, so we'll get started for today. So we were finishing up the Kalman filter last lecture, and I wanted to just do a brief overview of exactly how we go from the data in the R struct that you've been working on in homework 4 to your Kalman filter equations. And this will just be a high-level overview, but if any questions come up, feel free to pause or to ask questions. So we said we had this dynamical system. This dynamical system should actually also have the noise terms. And then we had our neural data, the Y's, which are that R.spike raster field, and our kinematic data, which are the R.cursor pause positions in the R struct that you guys have been working on in homework 4. And then what we said is we can fit the A and the C matrices by simply doing the pseudo-inverse, as we've done now several times in several different contexts. And then given that we calculated these A's and these C's, we could get samples of W and Q by just bringing AX, K, subtracting it and putting it on this side. And then we could use the formula to calculate the covariance of these W's and of these Q's like so. And so this was all pseudo-inverse and then plugging into our formula. After that, we know the parameters of our dynamical system. We know our A, C, W, and Q. And what we can go ahead is we can perform the Kalman filter recursion to calculate M1 and M2. And so this ends up being a while loop, as you'll implement on homework number 5, of these 3 complicated equations. And I should have mentioned at the end of last lecture that even I work with the Kalman filter all the time. I don't remember these 3 equations. And so there is some intuition in them, but it's not critical to actually implementing the Kalman filter to understand it. Really, the intuition that you want to understand is that after you calculate these recursions and you converge to your M1 and M2, then all your Kalman filter is doing is, as we mentioned before, it's weighting evidence from your previous decoded kinematics, which have information about the current kinematics, as well as the neural data, the Y's. And so homework number 5 will walk in detail through all of these different steps, and you're going to start off with that same R struct that you have on homework number 4, and you will build a Kalman filter from scratch. All right, so any high-level questions on the Kalman filter before we end the mathy part of the Kalman filter? All right, so now we get to finally see a video of the Kalman filter in performance. Oh. Okay. So here the monkey is controlling the Kalman filter. And you can see it actually has a bit of problems. Sometimes it fails, but when it gets to the target, it sometimes has trouble dialing in exactly to get to that target. And he'll take a few tries going back and forth before he actually acquires it. And so when this video is all said and done, the bit rate that we calculate is that he's at 2.2 bits per second. And if you will remember from the Wiener filter video I showed you, the Wiener filter was sitting at 3.8 bits per second. And so the basic vanilla Kalman filter that you might implement by just decoding the velocities that you measure from the training data actually turns out to not be as good as optimally weighting the history in terms of a Wiener filter. And so the question becomes, how do we actually get to the type of performance that we showed you before, the state-of-the-art performance that we call the refit Kalman filter? And so this algorithm was one that was developed in Professor Chenoy's lab. And it consists mainly of 2 innovations. And we're just going to talk mostly about one of those innovations today. And this innovation is something that we've only applied to the Kalman filter. You might think about applying it to the Wiener filter, and it might improve the Wiener filter even more. But for now, all we've done is quantified it with the Kalman filter. And so what this intervention is that we perform something called intention estimation on the kinematics. And so let's say that the monkey, for now, is just reaching with his hand from one target to another target. So he starts at a center target here, center, and let's say he's reaching up and to the right. If you actually look at the monkey's hand trajectory when he reaches from the center up and to the right, we actually might see a trajectory that looks a bit curved, as shown by this blue line. And so if you think about what you're doing when you fit velocities to this data, what you're doing is the velocities look like this, these green arrows here. And so when you take your first bin of neural data, even though the monkey has begun a reach to the upright, because of the biomechanics, he intends to reach upright, he actually makes a curved reach. And so your first velocity vector, instead of pointing upright, might actually point up. And so you can think of this as potentially some noise in the training data set. If the monkey was actually intending the whole time to reach just directly the upright, but he actually makes a curved reach due to biomechanics, then what we want to do is we want to regress not to this up velocity vector that we actually measured, but to this diagonal vector that is going up and to the right. And so what Vikash Gilja and Paul Nyujukian did in this algorithm is they thought of the idea that as you would in some control policy, you have some goal, and your goal is to always reach towards the target. And so what they did is they would rotate all of these velocity vectors to, instead of pointing in some direction specified by the green line, to always point towards the target. And it turns out that if you do this velocity rotation, you can improve performance. And so concretely in your training data set, what Vikash and Paul did is they took velocities, these velocity vectors, and they literally just rotated them so they pointed towards the target that you wanted to reach to. All right? And so in this paper, there's actually a bit of a different way that they do this. What they do is they perform 2 passes of training. And so in the first pass, they just train a vanilla Kalman filter from the hand data, as we've discussed. And what they'll do is then they'll run that poor decoder that you just saw in real-time control. And because that poor decoder makes many more errors, it would make errors as shown by this, this orange trajectory here. And for those errors, those are the velocities that they corrected. And so when you're making this decode, the cursor would jitter like this towards the target. But that error is introduced by imperfections in the decoder. And so what they did is they took all these velocity vectors and pointed them towards the target. And this is just a modification in the training set. So you can think of it as when you get your r.cursor pause from the R struct in homework 4, what you do is after you calculate those velocities, you just modify them a bit. And it turns out that doing this can really boost your performance. And so instead of that 2.8 bps, bits per second, that we saw in the prior video, what we can do is we can actually get that up to something like 4.5 bits per second. And so this is actually a very big modification. Any questions on this? It seems sort of intuitive, right? The monkey should always be aiming. His control policy is always to try to get to the target. And so this is trying to account for that control policy. Nathan?
Can you repeat again what modifying the pause data was?
So from the cursor pause data, what you did is you calculated velocities, right? Which were equal to r of some trial.cursor pause. So this is his cursor position at some time, we'll just call it I plus 1, minus the cursor position at time I divided by delta t. And so this will give you a velocity vector with vx and vy. And so let's say that we measured for this upward reach that vx and vy at the first bin was equal to 0 and 1. So instead of reaching up to the right, he reached directly up. Vy is positive, vx is 0. This modification essentially made this velocity vector equal to 1 over square root 2, 1 over square root 2. And so no longer was it a velocity upwards, but it was a velocity up and to the right.
You're modifying the actual cursor position data or just the velocity?
Just the calculated velocities. And so one other quick innovation that I wanted to talk about is we've mentioned that these systems are closed-loop feedback systems. And so the monkey, if he's controlling the velocity of the cursor, will see the position of the cursor update on the screen at each time point. And so in our Kalman filter, we had this term here, the covariance term, right, which modeled the covariance of my state estimate. It modeled the covariance of my position and my velocity. But this algorithm makes the assumption that because you see the cursor's position, that your uncertainty in the position should be 0, because you actually observe the position. And so I'm mentioning this because this is something that we'll explain on homework 5, and you'll actually program this 0 uncertainty in the position into your Kalman filter. Does that make sense? So normally the Kalman filter would have an uncertainty term for both your velocity and your position because you're drawing those velocities from a distribution, as we talked before. Now, because we actually show the monkey the decoded target position, the decoded cursor position, we make the assumption that he sees it, he internalizes it, and so he has 0 uncertainty about it. And so there's a way to put this into the Kalman filter, and you guys will see that in homework number 5. Ileana?
Can you, I guess, like know the direction if you're not looking for only one target? So like when you have the grid, how do you know that it's aiming for up and to the right if there's like targets that are just above it?
Yeah, so for the monkey, because we prompt just one target, we turn that one green, we assume that that's the one that he's reaching to. But then you're right that in the context of someone who is controlling a cursor to type on a computer screen, we don't know what target that they were intending to go to. So what we can do is we can post hoc look at the data, and let's say that they were trying to type out T, H, and E, the word the. But instead they typed out T, J, backspace, H, and then E. And so we know that ultimately when the participant was trying to control this computer cursor to type, she probably didn't mean to type the letter J because she then hit backspace. So then you can see, OK, well, if I go into my data post hoc, I can see that T, she intended to type. H, she intended to type. Actually, also backspace, she intended to type. And from that, I can know what she was intending to go to. And then post hoc, I can go ahead and rotate all those velocity vectors to point towards those keys on the keyboard. Anjali?
I'm sort of confused because if you're rotating the kinematics in the training set,
Yeah.
and when he's decoding, even if his thought process is going to make this curved path, then how is that going to hit the target when decoding?
Yeah.
Is his thought process I'm going to make a direct path, even though his arm says to make a curved path?
Yeah, so if his thought process was to make a curved path, then this correction here would worsen performance. And so what we're trying to get at is what is the monkey's actual control policy. And here we hypothesized that he was always intending to reach on a straight path or always trying to get towards the goal. And so when we programmed that in, we saw that it increased performance. And so that suggested to us that indeed this was his control policy, that he wasn't trying to command this curved path, but that at every single time point, he was just trying to get to the goal. All right, and so doing this change allows the Kalman filter to reach near native ARM levels of performance. And so here we have the monkey reaching to 8 targets, center, out, and back, exactly like in the R struct that we've given you. And on the right here, we show the velocity KF. And you can see it has some of those qualities that we saw in the video. Like when it reaches out to this target, you can see it overshoots it, and it has to come back to actually acquire it. Whereas if we do this modification in the refit KF, what we'll see is that he reaches to each target. He's able to stop precisely on each target to acquire it. And you'll notice that the variance in these trajectories is quite a bit less than the velocity KF. And the refit KF, just by eye, looks closer to the native ARM. And in quantifying performance, it's that way also. Gary?
So I see that right now it seems like the refit is making better progress because it's decoding intention. But I'm just wondering, the overshoot, is there any possibility that it's caused by the limit on operating, like the closed loop operating frequency it's too slow?
Yeah.
Is it more towards the intention that is the deciding factor in this case?
I believe that both of them are at play. So if you were to measure, and this is a measurement that we should do, if you were to measure the latency of the velocity kf versus the refit kf, I would expect it to be bigger. And what that translates to is in our equation, x hat k equals m1 times x hat k minus 1 plus m2 times yk. What that translates to is this M1 matrix being quote unquote larger. So if we just thought of these as scalars, if M1 was larger, then the previous history would have more of an effect. So I would expect this to be larger for the velocity kf. But then there's also an intention, a poor intention. So in the velocity kf, this is just anecdotal. You won't see this in any papers. When the monkey tries to stop on the target, because there's so much momentum in this M1, it actually overshoots the target, and then he has to go back. And so what we actually see the monkeys do is because they see this cursor updating, and they can change their strategy. What they'll do is when they try to reach these targets using this very poor velocity kf, when they get to the target, they'll start moving their hand in a circle. And moving their hand in a circle is just commanding all sorts of different velocities all over the place. And on average, this tends to cancel out to something close to 0, allowing them to hold the target. And so this is a strategy that the monkey developed due to the poor intention estimation of the velocity kf. Anjali?
Sorry, I think you just said this, but why does the refit common filter stop better? Why won't it still have the same inertia?
Oh, so because you've modified these velocity vectors, and so there's another modification that the refit Kalman filter does, which is when you are on the target, because you assume that you're always trying to reach the target, when you get to the target, it sets all the velocities to 0. And so that helps to dampen the inertia. Nick?
How long does it normally take the monkey to learn the new algorithm? I mean, like, are you able to control for the fact that he has more training on certain?
Yeah, this is a really great question. And the answer is that it depends on the algorithm. And so in Professor Shenoy's lab, we try to design what are so-called biomimetic algorithms, so that if you were to just reach with your natural hand and command those reaches, the decoder would try to match that as closely as possible. And for those, the learning time isn't long at all because he is almost controlling the cursor as if he would his natural hand. Up in Berkeley, there's a professor named Jose Carmina, and he's very interested in looking at how learning occurs in these systems and how neural plasticity occurs in these systems. And what he has done is he's given pseudo-random decoders to the monkeys where the weights are all intermingled. It's not completely random, but it's random to a large extent. And they see that this learning, the monkey can learn to use this decoder over the course of days. Now, the performance never reaches as high as what we can achieve with biomimetic decoders. But the monkey can eventually learn the mapping, learn some types of mappings. This is another topic for another time. But if the decoder is closer to what the native armor is doing, then he'll learn it in a matter of trials. If it's more jumbled, he'll take some days to learn it. All right, and so after performing all these modifications, what we get is performance that looks like this. Oh, there's no. So there's no sound in this video, but you can see the monkey reaches very straight to each target. When he gets there, he's able to stop on it and acquire it. And so this represents state-of-the-art performance. Gary?
I'm just curious about comparison. In this case, it's only showing one circle, but in the previous case, it was a grid. I'm just wondering if the grid might be offering more distractors than performance.
So what we've done is, and I will update this video for future lectures, but we'll put in a grid video. The monkey will perform this on a grid as well, and he will have the same level of control in spite of all those distractors. And so to rigorously compare, we of course do everything in the same exact task and quantify apples to apples the bit rate. Great question. All right, any last questions on Kalman filter? Nathan?
I don't want to take too much time, but you said there's this other step that when they get to the target, they set the velocities to 0.
Yeah.
In the case of not prompting a target, and say you have a target here and here and here, I guess you could say, okay, well, we know he will reach faster, and so the velocities will be greater if it's farther out. But are there any fine cases where that becomes a problem? Like, how do you know that he wants to reach for the first target instead of the second target?
So this is sort of related to Ileana's question. Ultimately, we just post hoc look at which targets we're sure the person controlling the cursor was reaching to. If we're not sure which target they reach to, then we won't perform either this intention estimation or this 0 correction. And so if they were typing THE and we knew that they reached to H and E, then for when he was over the H, we would set those velocities to 0.
Oh, so you can modulate how the filter itself performs. You could say, oh, I want to turn that part off. Like, I don't want to stop here.
This is all in the training data. And so it's not live online. So in the training data, what you can do is you want to try to make it as clean as possible. And so looking to the training data, we would say, OK, where are we sure the participant or the monkey was reaching to? And for the monkey, we're sure of all targets because we exactly prompt him. For the participant, we would pick the letters that make sense in the typing sequence that they made. And then for those specific trials, what we pick out, we would set those velocities to 0 when they're over that target.
OK. And
Yeah.
so you wouldn't do that in the actual testing when you're applying?
Oh, yeah. When we're actually running this closed loop, we can't make any modifications because that would be cheating. Right? If you have any knowledge of the task that they want to hold over this target and you set the velocity to 0, then you've used knowledge of the task, which for someone just using a cursor autonomously, they wouldn't have that knowledge. Yeah.
So if we're not doing like cursors to targets and we're, say, trying to implement a robotic arm or something, can you apply that technique, or is it?
Yeah, this is another great question. There's a lab at NYU PI'd by a professor named Bijan Pesseron, and I know one of his students attempted to do a refit type correction for a multi-degree of freedom arm. And what they found was that it's not exactly intuitive how to do it, because when you want to reach from this target to this target, there are many ways to configure your arm to reach for that target. And so you would likely have to incorporate other information into this intention estimation. So for example, you would want the trajectory to be a minimum energy reach. And when you incorporate information like this, you may be able to generalize this cursor goal correction to higher degrees of freedom. But no one has definitively showed it yet. Great question, Andrew. All right, so with that, we're going to move on to our last lecture, our last topic for this class. So throughout this class, we've been talking about how to interpret neural data, neural signals, to drive prosthetic devices. And so I wanted to recall the working definition of neuroengineering from Professor Shenoy's lecture, that neuroengineering is defined as the interdisciplinary field of both engineering and computational approaches to both problems in basic and clinical neurosciences. So far, we've been talking about applications to clinical neurosciences. We haven't talked too much about applications to basic neuroscience. And we'll start to uncover this today. Now, the techniques that we talk about today also have implications for clinical neurosciences, but they've been very fruitfully applied to basic neuroscience problems. And so in this lecture, we're going to talk about some computational tools that will both be helpful in understanding and designing neural prostheses, but also will be really useful for doing basic neuroscience. And so the reading for this lecture is first this paper by Cunningham and Yu, both of them former PhD students of Professor Shenoy. John Cunningham is a professor of statistics at Columbia now, and Byron Yu is a professor of EE at Carnegie Mellon. And they wrote this paper where they talk about dimensionality reduction for large-scale neural recordings. And this will basically be the focus for this class. I've also listed a paper here, which would be helpful to just read the first 6 pages to get an introduction into why we would do dimensionality reduction. And so imagine the scenario where we're recording from 20 neurons for T trials across 3 different conditions. The conditions can be different reaches. So a reach to the right, a reach up, and a reach to the left. And what we can do is we could plot spike rasters, shown here, these boxes. We have 20 neurons. We have 3 different conditions, coded in red, green, and blue. And we have many trials from 1 to T. And now the question is, we're looking at this population neural data. We have way too many spike rasters to look at. And what we want to do is we want to do some basic neuroscience. We want to understand what is the neural population actually doing here. And so the big question is, how do you find any meaningful structure in all of this data? Right? Just looking at it, it's hard to see because there's so much of it. You have also different conditions, different trials. On different trials, the activity looks different because of Poisson noise. And so this problem is getting immensely complex. And so what we're going to talk about in this lecture is a way to actually look, or to take all these spike rasters and pare it down to something simple that you can visualize to gain some scientific insight into what the population of neurons is doing. And so let's just imagine a very simple case. Imagine that we recorded spikes from just 3 neurons. And so we had the spike train for neuron 1, for neuron 2, and neuron 3. One intuitive way that we might think to visualize their activity is to take the bin spike counts of each of these neurons and plot them. And so very concretely, in this 3D plot, what I'm plotting are the bin spike counts for neuron 1 on this axis here, the x-axis, the bin spike counts of neuron 3 on this y-axis, and the bin spike counts of neuron 2 here on this z-axis coming out of the board. And what we can do is we can define a bin width. Let's say the bin width is 25 milliseconds. And so we'll take our first 25 milliseconds here, count the number of spikes for neuron 1, which we know is an estimation of that neuron's firing rate, count the number of spikes for neuron 2, and also for neuron 3. And what we can do is we can plot that as a point in space. All right? Is that clear to everyone, how I went from these 3 spike rasters to that 1 point in space? All right, and so we'll do the same thing for this next time bin. And this time bin will give spike counts for neuron 1, neuron 2, neuron 3. And let's say that that's this point in space. And then we'll do one more, this bin from here to here. And now that corresponds to this time point in space. So all we've done here is we've taken our spiking activity from these 3 neurons, and we've now found a way to just visualize it as a trajectory moving in 3D space. And so as time moves on, this trajectory moves along this, like so. Any questions about that? All right. And so what happens if we have more than 3 neurons? So in the data set that you're working with, we have 100 neurons. In the current formulation, what we've said is that each neuron is its own dimension. And as soon as we have 4 neurons, 5 neurons, we can no longer use this approach anymore. And so inherently, the problem is that we can't visualize more than 3 dimensions. And because this data is high dimensional, if each neuron is its own axis, is 100 dimensional, how do we visualize this activity? And so the key insight is that it may be possible to find projections of the activity where the neural signal exists and just look at those dimensions. And so what do I mean by this? So we're going to take our 3 neuron example where we had this noisy trajectory moving through space, which was just the bin spike counts at every single time bin plotted as a trajectory. What we can do is we can just smooth the data via convolution with a Gaussian kernel, as we talked about in handouts 7 and 8. And what we can do is we can see that even though this is a trajectory in 3D space, I can really find a plane where in this 2D plane defined by S1 and S2, I've captured the meaningful information in this trajectory. And if I were to just plot that in 2D, you'll see that the main features of the trajectory, that it first deviates this way and then comes this way. And for example, this green starting point is very different than this purple starting point, is all captured in this 2D plot. So this purple starting point is very different than this green starting point. And we've kept all this information about what the trajectory looks like. And so inherently, through this example, what we've seen is we've gone from a 3-dimensional system to a 2-dimensional system. And so now the setup for the neural data is, can we go from 100 dimensions to maybe, say, 10 or 20 dimensions, or maybe even fewer? And then can I visualize just the top 3 dimensions and get a really good look at what those 100 neurons are doing? Okay. Any questions on this? All right. So this technique is a technique called dimensionality reduction. And before we go on into the derivation of a way to perform dimensionality reduction, what we're going to do is we're just going to look at a few examples. And so this is a paper by Churchland et al. And in this paper, we have here a video corresponding to a monkey making a delayed reach task. And so this is exactly the task that we saw from handout 10 when we did communication prostheses. And what's going to happen is the monkey is going to be holding a center target. And then after some time, a target is going to appear. And we're going to see these neurons evolve. And when they turn blue, that means the target has appeared. After that, the monkey will then make a reach to this target. And his cue to reach to that target is given by the go cue, which we will indicate by these trajectories turning green temporarily. And so each of these trajectories corresponds to the population activity, the neural population activity, projected down into just 2 dimensions. And we're showing you the activity of all these trials through time. And so for now, there will also be a trajectory highlighted in red, and I just want you to ignore that for now. And so you can see the activity is evolving the baseline. It just got the target cue when things turned blue. And then when things turned green, he got the go cue. And then you can see all the trajectories seem to follow, all the different trials seem to follow the same trajectory through time. And so what we see here is that for all these different trials where the monkey reached up and to the right, or planned to reach up and to the right, and then made that reach, we see that there's similar structure in the population activity, where it starts off in some baseline state. Then when the target is cued, they converge to some state, maybe here. And then when they got the go cue, they all moved along a trajectory like this. Now, this noisy red trajectory that you see here is actually an aberrant trial where the monkey performed this reach more slowly. And because you can see that this trajectory for this one trial seemed to take many deviations, if you had to just look at this neural data, you would guess that that trial was the one where aberrant behavior occurred, and that is indeed the case. Right? So these techniques can also reveal interesting structure in the population. And so using techniques from convex optimization, what we can do is we can plot these 2D trajectories, again, in a 2D plane. But then what we can do here is we can start to rotate what these planes look like to find structure in the data. And so here, this is a technique that was developed called JPCA. And it looks for rotational structure in the data. And so the goal of this was to find these planes such that we saw rotations. And so we'll see some videos of them finding these rotational planes. And so you can see in these planes, the data originally looks very messy. But if you find the right planes, you can see that there's really interesting structure in this activity. And what this led to was the insight that these neural populations have really interesting temporal structure through time. And so you can think of our Kalman filter example where we said the kinematics at time k minus 1 inform the kinematics at time k through laws of physics. Well, this study proposed that because the neural data has interesting structure, as you'll see in this video, maybe the same types of dynamics, the same analogy of dynamics exist in the motor cortex. And so you'll see these are trajectories of different reaches through time. And you'll see that they follow these nice rotations. Question?
Could you explain a little more about how you develop those latent dimensions?
Oh yeah, that'll be the topic of this entire lecture. These are just examples that we're using to motivate it. But the rest of this lecture will tell us exactly how we find these latent dimensions. And so I just want to show two more examples. One is for decoding. And so if you'll remember for the planned reaches, what we did is we decoded the plan activity by integrating in some window tint. And so if you have the monkey plan reaches to 8 different targets, and you color the neural activity in that tint differently for those 8 different targets, and you plot them in a 3D space, what you'll notice is a topographical organization. So this right here is a plan to the reach, to the target to the down right. This might be a reach to the right. This might be a reach upwards, and this a reach to the left, for example. And so you can see that the neural population structure in these latent dimensions does reveal interesting things about the behavior. And in our last example, what we can do is we can actually use this to also visualize changes in the monkey's plans. And so in this task, what happened is a monkey was performing the same delay reach task that we've talked about. We prompted one target. The monkey planned to that target, and then eventually made the reach to that target. After that, what we did is we prompted a different target, say up and to the left. And then while he was planning to this target, we then changed the position of that target to be up and to the right. And so the monkey changed his plan from this target to this target. And he eventually makes the reach to this target. And so you're going to see how this is represented in the latent states. So this is the first reach just to the up right target. The monkey plans and then makes this reach. Now, this is him planning to the up left target. Then the target is switched to the up right target. You see he goes to the same plan state and then eventually makes the same reach. And so through these dimensionality reduction techniques, we can also begin to visualize changes in the monkey's plans. All right, so this is an approach inherently called dimensionality reduction. It's an immensely powerful tool, even outside of neural applications. So in applications where you have many sensors, you have very high dimensional data, these techniques can elucidate data that is inherently high dimensional. And so a fundamental assumption required in dimensionality reduction is that your dimensions are correlated. Because if your dimensions aren't correlated, then there's no way that I can project the data down in a way that captures all the activity from these different dimensions without having each of those dimensions be its own separate dimension. Does that make sense? So if the data is correlated, sorry, let me just plot a picture. If we have here neuron 1 and neuron 2, if the data looks like a line going like this, then knowing neuron 1 basically tells me all I need to know about neuron 2. If neuron 1 is firing high, then I know that neuron 2 is firing high. And so the system is inherently a one-dimensional system. However, if instead my data points look like this, then knowing what neuron 1's firing rate was, say neuron 1's firing rate was this value, I really don't have any information at all about neuron 2's firing rate. Neuron 2 could be up here or it could be down here. And so these purple dots here would be completely uncorrelated. And so we couldn't reduce its dimensionality. But for the red dots up here, they were correlated. N1, when N1 increases, N2 increases. And so because of that correlation, we could reduce the dimensionality. And so a thought experiment is, should the 100 million neurons in motor cortex be correlated? Right? Who says yes? And who says no? All right, someone who said yes want to venture an explanation? Nathan?
Your body has to move a lot of things together to achieve anything, and so the motor cortex should if it's trying to achieve something, it has to work with a lot of neurons, and so you have to have a lot of neurons working in concert.
Yeah. So there are a few things encompassed there. So hey, did you also want to offer up an answer? Or was that similar? Yeah, so Nathan said that your body is carrying out these actions which ultimately have synergies. And so the neurons in your cortex you have 100 million neurons driving three-dimensional kinematics, for example. And so there has to be some redundancy in this encoding. And for that reason, the data should be correlated. Okay? And so we're going to look specifically in this lecture at a technique called principal components analysis for finding these latent dimensions. And there are other techniques which are covered in other classes like CS229. There's an important technique called factor analysis. And then in EE378B, they cover so-called nonlinear dimensionality reduction techniques. And so we're going to be talking about PCA, principal components analysis, which is linear. But there are nonlinear manifold techniques, which could be useful if your data doesn't lie in a plane, but instead lies in some complex manifold. All right, so what if the signals are correlated? What is the dimensionality of this data? So you can just imagine we have some general x-axis and some general y-axis. And as we discussed, the dimensionality of this data is 1, right? I could fit a line through this. And what we could do is we could then project the data onto this line. And so this operation I've just shown here with the blue dotted lines. And so all I've done is taken the orthogonal projection of these data points onto this blue line right here. And if I were to then take away my red points, what I would see is that this blue line, which is one-dimensional, right? I could just redraw this as its own line and plot my orange lines on here, and I have a one-dimensional system. Captures most of the meaningful structure in this data. And so what do I mean by that? So one feature of this data was that this red point here that I've circled in purple is very different from this green point here. And there's a great distance between them, for example. And in my projection, I've captured that these 2 points are indeed very different along this line. And so this is what the projected data will look like onto principal component 1. And we've reduced the dimensionality from 2 to 1. Anyone have questions on this? All right. But the projection is really important. So what if instead of picking the line that we just picked, we picked instead this line right here? Now, if I project the data onto it, we'll see that, 1, the meaningful information in this data is squashed. And so we said that this point and this point were very different. And so if I were to circle this point here in green and then look at its projected value, it would be right here. And if I were to circle this point in purple and look at its projected value, it would lie right here. And so this axis doesn't really capture how these dimensions capture the meaningful structure in this data. Furthermore, if you just look at the spread of these orange points, you'll notice that it's very little. There's very little variance along this line. And so this projection isn't capturing the meaningful structure in this data. Anyone have questions on this? All right. So the goal of PCA is to find these good projections, which both minimize projection residuals. And so the projection residuals correspond to these blue lines, right? If these blue lines are smaller, it means that there was less error in my projection. And that's a good thing, because if there was less error in the projection, then I'm more faithfully reconstructing the original signal. And at the same time, it maximizes the variance of the data along the dimension. So what we're just talking about is the spread of points along your line. If you projected all of these data points and you ended up with a projection that looked like this, that had very little variance, you've lost all your signal information. You've projected it away. And so what we want is for these projections to really capture a lot of variance in the data. Okay? Does that make sense, both of these optimizations? All right. And so principal components analysis is a way to find these really good axes. And what we're going to show is that these two objectives that we had of minimizing the projection residuals is actually the exact same objective as maximizing the projected variance in the data. All right. And so what we can do is we can start to formulate this. And so let's say PC1 is this blue line right here. And so PC1 is a vector. We always make these unit vectors, so they have norm 1. So PC1 here would equal 1 over square root of 2, 1 over square root of 2. Now, if I were to take the activity xi and take the dot product, we know from our vector calculus, or just our our first vector calculus class, that the distance from here to here, the projected value, is just PC1 transpose, so the dot product of these two vectors of PC1 and x1. All right? And so if I wanted to know what this actual vector here that points from the origin and goes to this orange line is, if I want to know what the orange line's 2D coordinates were in space, then that would equal PC1 transpose times x1. That's the length of this from 0 to the orange point. And then I would multiply it by PC1. That would give directionality to my vector. And there shouldn't be a dot there. That implies dot product. We would just multiply it by PC1. And so this term here, PC1 transpose times x1, would exactly define this vector that I'm going to draw in green to this orange point right here. All right? Any questions on that? All right, so we'll finish with the next slide. What PCA, the cost objective that we're going to solve that will give us these dimensions u, is exactly minimizing this u, subject to it having a norm of 1. And so that was just to say that these PCs always are unit norm vectors. We want to minimize the norms of the residuals. So we want to minimize this dotted blue line here. And what is that dotted blue line? That dotted blue line is the vector xi, here shown in green, minus the vector here to this orange point. And here u is equal to PC1. And so this here is equal, the green vector here is just xi. And then the purple vector here is u transpose xi. That's the magnitude of the vector, times xi. And if I want to take the difference, which is here shown in orange, I would just subtract them. Right? And then if I want to minimize the distances, the size of these residuals, then what I could do is I could just take the norm of this vector and just square it. We like to square things because that makes these problems convex. And so what I could do is across all my n data points, and so this is for xi, but we have n of them, I could just continually calculate these residuals given by this term right here. And I could average all those residuals. And that would give me the sum of the squared residuals. And our goal is to find the u that minimizes this cost. And in the next lecture, we'll go into detail about how you find this u. All right? Thank you.